So, welcome to lecture 12. We're going to take a look at a bit more of the target responses of hormones and their effects on that target, right? Because we talked about, different types of hormones, different names of hormones. But now let's try to understand how they have their effect. Um, and lastly, we'll talk about stress regulation and how your body decides to summon you into a stressful situation or give you a call. So the first thing here we'll take a look at is hormone action. All right. So a few things about hormones. Um, First of all, what are the parts of the endocrine signaling cascade? We call endocrine signaling a cascade because oftentimes, it's not like those neurons where you send one neuron somewhere, release a bit of neurotransmitter, right? This is a hormone that gets released into the blood, goes all over the body systemically, binds to the target, has an effect on the target. Intracellularly, there's all these cascades, and we'll talk about that. So the parts of the cascade over here, right? We have this secreting cell, and a hormone, the little purple dot, is made by the endocrine gland, and it gets released into the bloodstream. We talked about this, and it binds onto this purple receptor, right? Over here. You see that over here, the little circle binds very nicely to the receptor on the target cell, right? So let's say this is testosterone. We know testosterone has, causes different things in the body. For instance, during puberty, it causes hair growth. So maybe this is a little hair follicle cell, right? And that has this receptor, and testosterone binds on it to create changes. So we call this the target cell if it's affected by that specific hormone, right? If it could bind to its hormone receptor on the target cell. I want to emphasize that look at the hormone and look at these receptors. This shape and the size is perfect for the two of them, right? This is called the lock and key fit. The key fits just in that one lock, right? So this is how there's a specificity in the endocrine system. Testosterone binds onto hair follicles, but I don't know of testosterone changing your, epithelium, right? Your little epithelial cells don't change due to testosterone. So let's say this is an epithelial cell. It has receptors too, right? Like look over here, it has this little orange one and it has many more, but it doesn't have this perfect locking key purple one that would bind testosterone. So testosterone will go near this epithelial cell. It'll go near every cell since it's in the circulation, but it won't bind onto this non-target cell, right? So all this to say, hormones, right? They bind to their target cell. They could have many target cells, right? But they also have cells that they don't target. And so, how do you differentiate these two? How does the hormone know to bind here but not there? Right? It knows that because of its receptor. The perfect matching of its receptor, if it's expressed on a target cell, then it'll bind. If it's not expressed, like this non-target cell, then it won't bind there. These hormone receptors, right? These purple ones where the hormone binds, they could be in different locations. They could be on the cell membrane. In this case, you see that it's on the cell membrane, but they could be in the cytoplasm as well. They could be on certain organelles. We'll see the mitochondria, and they could be in the nucleus. Okay, they could actually be near the DNA. We'll talk about this a bit further. What effects can hormones have on a target cell, right? We say, okay, the hormone goes and affects this cell. How does it do that, right? Its effect can be on the genetic material, the DNA. They could help produce a new protein by maybe activating gene expression, right? Remember that DNA is all the instructions to make proteins, and proteins are the workers of the cell. So these little hormones, right, could cause a cascade or could eventually end up binding onto the DNA itself and directly interacting with the DNA to start helping that gene get expressed more transcription and translation, right? It'll bind onto the DNA. It could also instead not even influence the DNA, but it could influence, the transcript, the RNA transcript, right? So they could influence the synthesis of a protein at this transcript level by blocking or allowing its translation. Maybe this hormone ends up causing a degradation of an RNA, or maybe it causes a blocking of an RNA, right? And lastly, it could influence at the protein level, right? It could actually go and affect the proteins that are being made. So, for instance, it could influence permeability. Remember, imperméable means not to let through the rain. Um, permeable, perméable means to allow it to enter the cell, right? So it could influence permeability of a cell by activating or deactivating a protein, right? What proteins allow permeability? What proteins allow things in and out of the cell, right? Well, we know that channels allow things in or out of the cell, right? Enzymes could maybe have an effect on getting rid of these proteins, or exocytosis of proteins, right? Maybe sending things out by these little vesicles. So over here, I guess the point is, for instance, let's say you're trying to control sodium ions, right? Let's say a hormone is trying to control sodium ions. The hormone could come in, have an effect on making, making this channel go onto the membrane. That'll allow sodium ions to flux in a bit more, or flux out a bit more, right? So we could have different situations where the protein level could be affected. Hormones could stimulate target cells all over the body. I really want us to understand this. Many cells around the body could have the same receptor that binds the hormone, right? And each cell type will have its own response. For example, many cells of the body respond similarly to glucagon. In the liver cells, right, in these hepatocytes, there are receptors for glucagon, meaning that glucagon that gets secreted by the pancreas into the blood can then go bind onto these receptors on the liver, right? And that binding in the end causes the breakdown of glycogen into glucose. And it's also, in addition, starts gluconeogenesis, right? So over here you see liver cells start breaking down glycogen to glucose to increase the glucose in the blood. In muscle cells, right, it's a little bit different. It causes the breakdown of glycogen for self-use, but it's still binding and having kind of a bit the similar effect of going to break down glycogen to glucose, but the muscle uses it itself. Do you see how the liver has receptors for glucagon and skeletal muscle has those same receptors for glucagon? And therefore, glucagon has the same kind of effect on both of these tissues, right? It gets more complicated than that. There can exist more than one type of receptor type on different target cells. For example, let's consider epinephrine, right? Over here in liver cells, hepatocytes, epinephrine, right, that stress response, remember, like epinephrine is this adrenaline, right? So when you're under the sympathetic response of epinephrine, you want to put more glucose into the blood. So it causes the breakdown of glycogen into glucose. Here it is. Epinephrine in red binds to these receptors, these beta receptors on liver cells. And that causes the glycogen deposits to be broken down and released into the circulation to put more glucose into the blood so that you're having some glucose as potential energy to take in to your cells, right? So these reserves in the liver cells are being broken down. We know that, right? This is exactly what we were talking about over here with the glycogen breaking down to glucose. So we know that epinephrine has that effect. And here in this picture, we see that it's due to the beta receptors, right? In smooth muscle cells, epinephrine also has effects, right? But smooth muscle cells are not responsible for breaking down glycogen, right? Smooth muscle cells have a different cellular response, right? They're there to kind of constrict or relax, right? And could change the size of the vessel wall. Interestingly, we have the epinephrine binding to the same beta receptors here in the blood vessels of the skeletal muscle, right? And we know that we want to have more blood flowing to skeletal muscle when we're under stressful conditions. So this epinephrine binding allows those intracellular proteins in the smooth muscle to dilate. So here we have a dilation effect. It's a different cellular response because these are different types of cells that are being bound to. And inside, yes, it's the same receptor, but inside we have different mechanisms for hepatocytes versus these smooth muscle cells. Now, over here is very interesting. When you're under a stress condition and release epinephrine, you don't want to have blood flowing to your gut, to your intestines, right? Resting and digesting, that's for a relaxing period. So when epinephrine binds, right, to have its different effect on this same type of cell, right, this blood vessel that's elsewhere, the intestinal blood vessels have actually a different receptor, these alpha receptors, that end up having the opposing effect when epinephrine binds, right? So these vessels will constrict. And so that's how, when you're stressed and you release this epinephrine has so many effects, but it could affect the tissues differently depending on what receptors are expressed, right? I guess that's my main point here. When you release a hormone, right, it's not going to release glycogen in every single tissue that it binds to. No. Yes, in the liver cell, it'll cause that because that's the specialty of that cell. But then in something like a vessel, right, it'll end up having its effects there based on the intracellular proteins that are there. And secondly, depending on what receptor epinephrine binds to, epinephrine could bind to a couple different receptors, it might do the complete opposite effect as the same tissue somewhere else in the body, right? So receptors are important as well. Okay. The next part to talk about is lipid soluble and lipid insoluble hormones. I want to get this terminology straight. So if you prefer the words that I drilled into your minds in the previous course, lipid soluble, to be soluble means to mix well with, right? If I mix well with lipids, this is called a hydrophobic molecule, right? Hydrophobic. If it's lipid insoluble, right, meaning it's not soluble in a lipid, then this is a hydrophilic molecule. And I put them in different colors to help you out a bit. Blue mixes well with water. Yellow is like fat. It mixes well with lipids, right? So lipid soluble or insoluble, how do hormones interact with the target cell? Right? When a hormone binds to the target cell, how does it do so? Well, it depends on their hydrophobicity. It depends whether they are lipid soluble or insoluble. We're going to see that there's two different kinds. So we categorize hormones according to this property. Lipid insoluble, meaning that they are not soluble in the lipid, right? This is called a water-soluble molecule. It mixes well with water. You see the molecule splashing in the water. How does it get transported? It's dissolved in the blood, right? This could easily move around in the blood. It likes the water. Water, we know that blood is watery. The cell membrane, right? When it gets to the cell, it is water loving, so we know that hydrophilic molecules do not cross the plasma membrane, right? So they cannot cross this cell membrane. And instead, we have receptors that are located on the cell membrane. So there's low receptors on the cell membrane that are waiting to detect it. That's exactly what I was talking to you about before, having low receptors on the cell membrane. Examples of this, non-sex hormones. So we're going to see that the sex hormones are mostly the ones that are lipid soluble. Over here, for example, right, you make a little hormone from the adrenal glands, you send it into the circulation, the little veins, and then once it gets to its destination, it binds onto a receptor that's on the cell membrane. These are hydrophilic, water-loving molecules, right? Philic means loving. Lipid soluble hormones means they are soluble. When something's soluble, it mixes well, right? Lipid soluble hormones, right? These are fat soluble, meaning they could mix well with some fats. They are transported bound to a transport protein. They do not like to be floating in the blood. That is very watery. So a carrier protein or a transport protein binds to it to allow it to move through the blood. The cell membrane, right? These hormones can cross the cell membrane. And therefore, the receptors, they are not on the cell membrane. Look at this arrow. It goes into the cell. Oftentimes they're inside, either in the cytosol or the nucleus. So this is very interesting because all of a sudden we don't need to put receptors on the membrane. They could be on the inside, right? We always think of receptors on the membrane, but in this case, the receptors could be floating around in the cytoplasm. So again, these are like sex hormones, right? Estrogens are an example of lipid-soluble hormones that get carried around in their little boat and then sent into their target cell. These are hydrophobic, water-fearing hormones. So now I'm going to break down. We're going to look at lipid insoluble and lipid soluble. So lipid insoluble, right? If it's water soluble, in other words, and it could actually go and it likes water, then we're going to see that there's a few different types. We're going to see amino acid hormones, peptide hormones, and protein hormones, okay? All of amino acids are the single little monomer, right? Peptides are a little bit bigger, and proteins are very big, right? All of these, what do they have in common? They're proteins, yes. We call these the protein hormones altogether, but what's special about them? Proteins. Remember, these are polar molecules, right? They are hydrophilic. They love water. Fat does not like water, right? But these are all hydrophilic. They like floating around in water in the blood vessel, right? So you see these little gray hormones just floating around in the capillary, right? In the blood vessel. They love it. Travel, right? These hormones, these hydrophilic, travel freely in the blood since it is a hydrophilic molecule. The mechanism, right? They diffuse out of the bloodstream and bind to receptors. You see the brown receptor on the targets, and they act indirectly on the cell through a domino effect called the signaling cascade. We're going to see this in detail, but look at this. They cause all these little proteins to move around, converting stuff. This protein binds to that one binds there. You make this thing, this other thing activates the other protein. This is called a signaling cascade, and it's indirect because the hormone doesn't directly affect the outcome. It starts a cascade of domino effects, right? We're going to see how this is a bit different for the lipid ones, the lipid soluble. Time of onset, these hormones act faster than lipid soluble hormones, right? But the response time, a few minutes to less than 1 hour. These are their response time, which is a bit shorter, right? We'll talk about that a little later. So how is the signal perpetuated in the cell? Let's go through the nitty gritty over here. So step 1, the lipid insoluble hormone, we call it the first messenger because it's the first of the cascade of the messengers, right? I'll show you why that is. This first messenger binds to the brown receptor here, right? That binds it specifically, just that hormone. Recall these hormones cannot cross the plasma membrane, right? The receptor, the brown receptor here, activates the peripheral membrane proteins. Remember, there's proteins on the peripheral side sometimes, right? These are called peripheral membrane proteins. And these proteins are called G proteins, okay? These G proteins help, what's the point of this? Is so that there's some crosstalk between the outside world and the inside world, right? So these are called G proteins, right? And they kind of help, in a sense, signal the other side of the membrane what's going on. Oh, hey, there's a hormone that just bound to the other side, right? So something will happen to these G proteins. So the G proteins activate when a phosphate group is added to a bound molecule of GDP to make GTP. So G proteins are just GDP and GTP, okay? It's kind of like ADP and ATP. Remember, when you phosphorylate something, you add a phosphate, it becomes very powerful, right? It becomes activated. So guanine diphosphate gets turned into guanine triphosphate 3, just like ATP, and it becomes activated, right? And this then activates an enzyme called adenylate cyclase over here. And anything with an ACE is an enzyme that makes stuff or breaks stuff, right? Adenylate cyclase makes AMP, adenosine monophosphate, right? So this cyclic adenosine monophosphate, CAMP, right, is made by adding a little ATP to that. Now, so this enzyme, adenylate cyclase, makes that CAMP, right, which is a second messenger. This second messenger molecule, right, is the one that will then go have its amplified effects. So basically, we make a lot of this CAMP, right? And then this second messenger, right, can then turn on other enzymes like kinases. Okay, so here we're going to activate some kinases. Basically, it's allowing other enzymes to be turned on, or it might even open ion channels to cause a response in the target cell. So it might go and open up some little channels. And then other enzymes get phosphorylated, right? On and on and on. And you get this actual response that you want, okay? Now, this signal needs to stop. So step 5 is that other enzymes break down the secondary messenger to stop the response, making the response short-lived. We don't want this going on and on and on forever, right? Now you're going to tell me, what the heck is this first messenger, second messenger? Um, all that it is the fact that at each step here, there's an amplification process of the signal, since each messenger can talk to many other key players. Look here, this one little hormone, this one first messenger, right, binds onto one receptor, which then activates many adenylyl cyclases, which then create many CAMPs, which maybe makes all of these activate many protein kinases, which then have their effects on many other little protein molecules, right? So over here you see that it's not like one protein binds to this other one and the other one. It'll bind to many. It'll bind and bind, activate, inactivate, right? So the signal gets amplified, and I think that's the key response here. The key thing to keep in mind is that these first messengers bind on to second messengers and third messengers, and then you get this amplification process. Now, receptor types and secondary messengers. Remember epinephrine and norepinephrine, not only are they neurotransmitter, but they're also lipid insoluble hormones, right? We know that they get released from, they're a little bit of a weird one because they're a bit of both. They're a neurotransmitter, but they're also a hormone. Remember, when they get released, right, they bind onto these receptors. Well, it's the same thing in the hormone system, okay? So they bind onto these adrenergic receptors. Remember the alpha or the beta adrenergic receptors? Well, in this case, right, it makes sense. Epinephrine, I told you, didn't have the same effect everywhere in the body, right? In the blood vessels of the skeletal muscle, there was alpha receptors, whereas in the blood vessels of the gut, there was beta ones. So they have different effects, right? Let's go through those effects. When epinephrine or norepinephrine bind to the alpha receptors, right, the alpha 1 receptors, they can open calcium channels, right? And that causes some smooth muscle contraction in this region, right? So, for example, different depending on where you are in the body, this will cause this contraction. Over here, you see the calcium channels; they're open, right? Um, so that's very important. This alpha receptor, right? You end up getting some calcium channels open. If you look at the alpha 2 receptors, right, they activate enzymes to break down secondary messengers like CAMP. So alpha receptors, they inhibit the release of this calcium, right? And they inhibit this adenylyl cyclase, right? So over here, if you go back, they kind of get rid of this CAMP over here, and they stop this propagation. So this is great. If I have an alpha 1 receptor here, I could cause contraction, but if I have it at another location with alpha 2, then epinephrine at that location could cause the opposite effect, right? And beta receptors, right, activates CAMP as the secondary messenger. So kind of does the opposite. It really promotes the formation of CAMP, and you get this smooth muscle relaxation in this case, right? So we could these receptors have, you should see sometimes the cascades that are there. Don't worry about these. Just know I guess the key point is each receptor has its own kind of identity and its own effect, right? The Now we can start having this talk that the endocrine system is confusing. there's different hormones that do different things. Glucagon does this, insulin does the opposite. But on top of it, when one hormone, let's say just epinephrine, binds to a receptor, it can have different effects because it might be a different receptor. Epinephrine binds to A1, A2, and beta, right? But if one cell has just A1 receptors, it'll have one effect. If one cell has just beta receptors, it'll have a complete different effect. So not only are there many hormones, one hormone has many receptors that could have varying responses. Um, and this is why, just you don't need to know this, but this is why hormonal therapies work, right? Or try to work, because there are different types of receptors for drugs, and it's easy to target just those cells, right? Or there are some meds that just bind to that specific type of receptor, so it's a bit easier to target sometimes. Um, yeah. So I think the next two slides are repeating the same thing. It's just to kind of ground it in a bit more. So CMP is one of the most important secondary messengers there is, right? So I just want to go a bit more into detail that those secondary messengers, like CAMP, the red triangles here, often activates what we call these protein kinases. Kinases are proteins, they're enzymes because it's an ACE, right? And these things just add phosphates everywhere, okay? These are enzymes that can activate or inhibit other molecules by adding phosphates. You see this orange kinase just adds a phosphate over here, right? Or it just adds a phosphate over here. These are phosphate adding molecules. You'll talk about different kinases with drugs, okay? And these kinases are important to activate. In ATP, guys, what is so special about turning ADP to ATP? You're adding a phosphate. Phosphates energize things, right? So when a hormone binds, in the end, they just start energizing other proteins to do things, right? Maybe to change the DNA, maybe to break a transcript down, right? It depends. So I want to run through this one more time, right? Step one. In this example, you see the first messenger, in this case epinephrine, binding onto its receptor. In this case, it's a beta adrenergic receptor, right? If we go back, the beta receptors that are here will go and activate the CAMP, right? So these beta adrenergic receptors cause an intracellular G protein activation. Here you have these G proteins, right, that get activated, which eventually makes CAMP, right? They go to the adenylate cyclase enzyme and they tell it to make a lot more CAMP. CAMP is the second messenger, right? That this CAMP, which is now made, is going to go and activate these protein kinases. Specifically, we talk a lot about PKA, protein kinase A. This PKA, remember, a kinase is something that adds a phosphate, right? So this PKA is going to add and phosphorylate, remember that word, add a phosphate to a variety of proteins, right? Which will eventually trigger a variety of effects on cellular metabolism, synthesis of proteins, or secretion of products, right? Maybe this is, let's go through that bit by bit. When we phosphorylate proteins, maybe it'll trigger different effects on cellular metabolism, right? Maybe that protein will then go break down something to slow down metabolism. Maybe it'll affect synthesis of proteins. Maybe it'll make more proteins, right? It'll bind eventually in the nucleus and allow the DNA to change so that it makes more proteins, little channels to put in the membrane. Or it might affect secretion of products. Maybe it'll tell the cell to make these little exocytoses of different transmitter, Okay, why do lipid insoluble hormones cause indirect effects? So, I told you that these lipid insoluble hormones, the ones that are hydrophilic, right, they do not have direct effects. What did I mean by direct? Well, what I meant by direct is you're directly affecting the DNA, let's say, or you're directly affecting the mitochondria. In this case, hormones, they create a cascade, and then that will have some kind of target cell response, right? So let's go through that indirect pathway for lipid insoluble hormones, right? Again, I'm repeating this over and over. Hopefully, it sticks some way. The membrane receptor is always here, right? And this is our fancy schmancy hormone, the first messenger, we call it, either epinephrine, norepinephrine, or other peptide hormones. And these bind onto a receptor, a G protein receptor, right? The G protein is activated, right? DP to GTP. The G protein activates an enzyme, adenylate cyclase, that will make the second messenger, CAMP. The second messenger, right, CAMP, will go and activate kinases that will go and phosphorylate a bunch of things. Or it might open some ion channels on the plasma membrane that will let in some ions, right? And then these induce responses in the target cell, right? This is the desired response. For example, if you think of ADH or aldosterone, right? We want to go and change the level of ions or water in the cell, right? You might end up putting a channel at the top of that membrane, right? Lipid soluble hormones. Okay, so some hormones, I put this in yellow, right? Because the blue is for ones that mix well with water. They had this whole mechanism, but the lipid soluble hormones, right, are mix well with fat, yellowish stuff, right? So that's why I put this in yellow to remind us that this is lipid soluble. Um, the subtypes of these, right? Uh, these we mostly call them steroid hormones. We're gonna see that steroids are lipid soluble. Um, when they travel, right? They are bound. We're doing the whole story again, but for lipid soluble hormones, okay? So when they travel, they're bound to water soluble transport proteins. You see these blue things? These are transport proteins so that the free hormone could actually go and kind of move around. If not, it wouldn't be able to, and it would clog the vessel and cause atherosclerosis, where you have an accumulation of plaque. Um, Mechanism, right? They act directly with the intracellular receptors in the cytosol or the nucleus. Okay, so um, you see that this free hormone, right, gets released eventually into the cell, and that it passed directly into the cell, right? It didn't bind onto a receptor on the plasma membrane. It went directly inside, and they bind directly to a receptor. Here you see in red, right? In the cytosol, or sometimes over here you see it in the nucleus, right? Oftentimes, hormone receptor complexes, when they bind together to form a complex like this, they could interact with the DNA directly. This is why we say that these are direct mechanisms, right? It's not like it binds to a receptor on the plasma membrane, causes a cascade which then affects the DNA. Here it's going directly in, binding to the receptor and going onto the DNA, right? And they act a bit like a transcription factor, right? It's a factor that affects transcription. And it says activate this transcript more, activate this protein, activate this less, right? It could be either or. So these could directly trigger the expression of genes to synthesize proteins. Okay? Time of onset. it takes more time to act than lipid-insoluble hormones. So it takes a bit of while. Look at how it needs to travel in the blood, right? It needs to travel in these transport proteins. Um, and response time, they remain intact much longer than lipid-insoluble hormones, okay? Now, how is the signal perpetuated, right? Let's go through it bit by bit. So these hormones diffuse or are transported by proteins in the bloodstream, right? Over here you see them with the transport protein. Step 2, the hormones cross the cell. You see it over here. The cell membrane to bind to cytoplasmic receptors, or in this case, nuclear receptors. This is a nuclear receptor. It's in the nucleus. And they create a hormone receptor complex. You see one made right here. This hormone receptor complex can then interact with our DNA to alter gene expression and affect the production of this new protein that you're making, right? Altered gene expression increases gene transcription and making an mRNA, and then eventually it'll affect, right, that protein synthesis that you're doing. Why do lipid soluble hormones have a direct effect? Because they directly bind onto the DNA, right? Step 1, the lipid soluble hormone diffuses or is transported through the plasma membrane. I told you that the hormone will then bind to either a receptor directly in the nucleus, over here, 2A, or 2B in the cytosol to create a hormone receptor complex, which in the end ultimately moves to the nucleus and has its effect, right? So it could be anywhere inside the cell. Step 3, right? The hormone receptor complex affects gene expression by altering the DNA somehow and can alter the activity of that cell, right? This DNA then makes another protein which has its desired effect, right? Okay, so over here, let's look at a few questions. Insulin operates by activating a receptor on the outside of the target cell. Based on your knowledge of hormone types, do you expect insulin to be an amino acid based hormone or a steroid hormone, and why? Here it says insulin operates by activating a receptor outside the target cell. So um, I believe, right, it will not be a steroid. Steroid hormones would go into the cell, right? So it's not lipid soluble. So we could cross out B and C. Let's look at C. Sorry, we could cross out B and D, right? Let's look at C, amino acid based, because it is not water soluble. Amino acid hormones are water soluble. They mix well in water, right? So I think it's A, amino acid based, because it does not pass through the cell membrane of the target. It'll actually bind to the receptor on the outside, right? And let's look at this one. Why do steroid hormones use a transport protein? Transport proteins allow steroid hormones to cross the cell membrane, leading to a cellular response. No, the cell membrane is hydrophobic, and steroid hormones can easily cross if they don't need help to cross, right? Transport proteins stabilize the hormones so they don't react chemically with the blood. Not really. Transport proteins make steroid hormones temporarily lipid soluble. They already are lipid soluble. Or transport proteins make steroid hormones temporarily water soluble. Yes. So they temporarily help it be soluble in the water. Okay, hormone classification. So based on this same lipid soluble, insoluble, we're going to take a look at different types of hormones. Amine hormones or amino acid hormones, protein hormones, okay, and steroid based hormones. So the first category is these amine hormones. these are lipid insoluble. They're amino acids, so they can't mix well with lipids, right? So these will be floating in the blood, right? Um, a little exception is T3 and T4. Of course, there's always a stupid exception. So this is thyroid hormone is one of the exceptions. But everything else, every other amine hormone is lipid insoluble. If it's lipid insoluble, right? If these amine hormones are hydrophilic, then they act indirectly. They do not cross the membrane, right? They need to act on receptors on the outside and indirectly cause a cascade. The composition of these amine hormones, they are a single amino acid with modified groups. It could be either, the ones I know of are tyrosine, right? This is a tyrosine. You have the COOH over here and the N terminus, and this is your R group, right? This is a modified amino acid. Here you have the second one I know of is tryptophan. You have the carboxyl group here. You have NH2, the amine group, the acid group, amino acid, right? And then over here you have this ring structure, this tryptophan. This is the R group where you have a specific molecule, right? And this makes tryptophan. These are tyrosine and tryptophan are the two amino acids that I know of that will be the precursors to make the specific hormones, okay? So look at this. The examples we'll look at are derivatives of tyrosine. If you derive this and modify it a bit, tyrosine is not a hormone, right? But if you bind it to another one and make a few modifications, you get this thyroid hormone, right? Or you could get some catecholamines such as epinephrine, right? Over here, you have this epinephrine that is a modification of this tyrosine. How do you get tyrosine, right? You get tyrosine from meat, dairy, fish, and other stuff in your diet. And this tyrosine then gets converted by either the thyroid gland to make T4, right? Or by the adrenal glands to make epinephrine, okay? Tryptophan over here, right? If you modify it a bit, you get this melatonin, right? Melatonin, remember that's to go to sleep. Turkey is very well known for its source of tryptophan. Other sources include chocolate, oats, blah 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 blah. This is why they say, when we eat a lot of turkey during Thanksgiving, you get a bit sleepy, right? Because this melatonin is being made a lot. You get a lot of tryptophan from turkey. So again, these are derived from amino acids, so we call them amine hormones. I want to really emphasize we're in the blue right now. Like, this means that they're lipid insoluble. Amino acids are polar. Look at the polar group here, OH, NH. These are polar molecules, right? They're hydrophilic. They will actually go and dissolve well in water, right? Examples of those amine hormones: epinephrine or adrenaline and norepinephrine, noradrenaline are examples. The adrenal medulla, right, will take this tyrosine molecule and convert it into norepi and epi. Okay, precursor made from tyrosine, release by the adrenal medulla. Response leads to sympathetic activation, right? You activate that sympathetic nervous system. Over here, we have melatonin, right? Melatonin is, we talked about, made by the pineal gland. The pineal gland in the brain will convert tryptophan and convert it into this melatonin. What is its response? Remember, melatonin controls circadian rhythms and inhibits reproductive functions, right? So that's the role of melatonin. Um, amine hormones. Okay, so this is where it's annoying. Remember over here I said amine hormones, they're lipid insoluble, except T3 and T4. So unfortunately, thyroid hormone is annoying because it is considered a lipid soluble hormone. So let's go through this bit by bit. This is why I put it in yellow. Um, this amine hormone, right? The thyroid hormone, also known as T3 or T4. T3 and T4, sorry, these are two types of thyroid hormones. They're both made. The precursors, we know they're made from two tyrosine molecules, amino acids, right? And they require iodine atoms. It's two of these tyrosines plus a bunch of these I molecules, right? Iodines, released by the thyroid gland. What type is this thyroid hormone? Well, unfortunately, this is the exception, right? Although it is an amine hormone, which usually proteins are hydrophilic, right? Um, Although it is an amine hormone made from tyrosine, which is hydrophilic, it likes water, right? It gets modified into a form that is lipid soluble, right? It gets converted, right, into this crazy form and this crazy form that actually the properties of this make it lipid soluble, okay, and can cross the cell membrane to cause direct effects. So this is just an exception where this one actually goes into the cell and has its effects, even though it's an amine derived from an amino acid. What responses do thyroid hormone have? We know this. It is a systemic hormone and affects cellular metabolism of almost all cell types. They cause an increase in body temperature via a calorigenic process, right? We know that the thyroid hormone increases body temp, influences skeletal muscle and neuronal development, very sensitive to O2 and CO2, right? And can accelerate mineral turnover in bones. In other words, thyroid hormone affects metabolism, we say, right? Remember, it affects weight and all of these things, right? And growth. So that's the response. Because it's a stupid exception, I need to go through kind of what it does in the cell. But this acts very much like a typical lipid insoluble hormone, right? Step one, thyroid hormone are transported across the membrane because it's able to cross. The hormone will bind to a receptor that's either in the nucleus or, in this case, interestingly, in the mitochondria, 2B, right? Here it's the mitochondria. Remember, this is metabolism. It affects the amount of ATP, right? So it binds, in this case, on the mitochondria, and in some cases, it also binds on in the nucleus. The hormone receptor complex, right, when they bind together, they either cause a change in gene expression over here, 3A, um, which then alters the structure of proteins and has its target, or they bind, the ones that bind onto the mitochondria, they will increase their ATP production, right? And change metabolism. They'll metabolize a lot, right? So excess thyroid hormone will cause a lot of weight loss, right? Because you're burning a lot of those different molecules or nutrients that you have to make a lot of ATP. And step four, these will change the behavior of the target cell in some way, right? You get some that desired change in metabolic change. Okay, so that was the weird exception. So in general, though, amine hormones, they were, they liked water, they were hydrophilic, and they were derived from a single amino acid. Protein hormones, right, are not single amino acids, okay? They are bigger than just one single amino acid. These are all, thank God, lipid insoluble, meaning that they all will not dissolve in lipids. They will dissolve in water, right? That's usually the case for amine hormones, except the thyroid one. The mechanism still acts indirectly. It binds onto the receptor that's at the plasma membrane and has a cascade, right? Composition includes small peptide chains or large folded proteins. So sometimes we call them peptide hormones if they're 2 to 100 amino acids, or these proteins, right? Large folded proteins when they're more than 100 amino acids. Okay. All this to say that it is more than one amino acid, right? And since it's not just since it's more than one amino acid, it doesn't simply, get brought into the body. You need to create it. You need to synthesize it by a ribosome, right? You actually need to bring together and express this protein and bring together the amino acids to link them up. Examples of this, here you have oxytocin, this little peptide hormone, which is 9 amino acids long. Okay, here you see glycine, leucine, proline, cysteine, right? Here you have human growth hormone, right? This is 191 amino acids. This is one of those large protein hormones, right? Each of these is an amino acid that gets folded up. So example of some protein hormones, I just gave you a few. So oxytocin, right, was 9 amino acids long, stimulated by physical contact and influences emotional bonding, right, released during childbirth and lactation. Here you have ADH, antidiuretic hormone, anti-diarrhea, meaning you don't want to put water out of the body, you want to retain that water, right? So this is also known as vasopressin in clinic. Vasopressin, vaso means blood vessel, pressin means pressure. So remember, getting rid of water or keeping water in your vessels will affect your blood pressure. So that makes sense why it's called vasopressin. This is called ADH, right? These are 9 amino acids long as well and regulates the body's water balance by controlling how much water is reabsorbed by the kidneys or put into the urine, right? So this simultaneously helps maintain blood pressure and blood volume, all right? Hormones released by the pancreas, we have insulin over here. Two chains linked together, the alpha chain, that's 21, and beta chain is 30 amino acids. They're linked. This is what insulin looks like. Glucagon here is 29 amino acids long. These hormones regulate blood glucose levels. Insulin signals for glucose absorption into cells. Glucagon signals for glycogenolysis, breakdown of glycogen lysis, lysing the glycogen by the liver cells, right? And so those are some examples of those protein hormones, right? Last but not least, we're not in the blue anymore. We're in this yellow zone, which means that we are lipid soluble. We like to break down in lipids where we like lipids, right? So we do not like water, right? Remember, these are hydrophobic. So the mechanism, it acts directly. Remember, the complex will bind usually directly to the DNA. So they are bound to transport proteins like albumin in the blood because they cannot go and just go around the body, right? They need to be bound to some proteins like albumin. This extends the half-life of steroid hormones, 60 to 90 minutes for steroid hormones versus 1 minute for amine. So the fact that they're bound to these albumins actually helps them to stay around a little longer, which is why steroid hormones oftentimes have this extended lifespan and therefore they could act for longer in the body. Composition, It's made from cholesterol. This is cholesterol over here, and we're going to see that many of these steroid hormones are from this cholesterol molecule. Okay, so examples of some steroid hormones. We have cholesterol makes this molecule, and then it makes testosterone. Testosterone is released by the testes, right? Uh, if you add one little hydroxyl and molecule, then you get estrogen and progesterone, right, which is down here, that are released by the ovaries. If you take this precursor and instead put a few other groups, right, then you get glucocorticoids such as cortisol, right, that are released by the adrenal cortex. These are stress hormones involved in glucose metabolism. And if you turn progesterone and add a little other molecule, right, then you get aldosterone, which is a mineralocorticoid released by the adrenal cortex, and regulates sodium and water reabsorption in the kidneys, right? Okay, this is pretty much it for the chemical classes. There's one type of steroid hormone I kind of forgot to talk about, and it's just a little sidebar. It's another one of these lipid soluble ones, but it's not steroid ones. These are called fatty acid based hormones. Okay, so these are largely lipid soluble but act indirectly due to their slightly polar properties. Okay, so they actually bind to G protein couple receptors since they have some polar properties. You see in green here, you see that it's negatively charged, so it's polar. These are also called eicosanoids. Eicosanoids, and so they're derived from this arachidonic acid, which is an from the omega-6 fatty acids. Um, so these are known to be called paracrine hormones because they influence cells of the same tissue. Not long distance hormones, right? So they influence themselves, kind of, right? So over here, we have a few examples, three. So we have arachidonic acid will make prostaglandins, which have a role in pain and inflammatory response and fever. Thromboxanes, right? Sounds like thrombosis, so clotting. So they have a role in blood clotting. And leukotrienes, right? Mediate immune response as during inflammation. Okay, leuko sounds like white blood cells, right? Inflammation, we know there's a link there. Um, and now, just to finish off a few things, right? So, hormone transport in the blood. We talked about this a bit. How are certain hormones transported in the blood? Lipid insoluble hormones, hydrophilic hormones like epinephrine and norepinephrine, and any of the pituitary hormones are soluble in water and do not require any transporters in the blood. Lipid soluble ones, right? Over here you see this little hormone like T3, T4, and all the steroid hormones. They require plasma globulins and albumins, these bound carriers, to carry them and transport them in the blood, right? Okay, hormone interactions. So this is pretty cool. And it complicates our life even more. Like, we know that there are many hormones. And now we know that even for that one hormone, there's many receptors, right? But on top of it, every one cell could have many types of receptors, right? And activation of different receptors activate different enzymes and open different ion channels, okay? Each cell can have various combinations and combine different hormone signals at once, right? Here's your little cell that has many, many, many types of these receptors, right? When multiple hormones bind to the target cell simultaneously, their effects could interact, right? So some hormones, we call two hormones synergistic hormones if they stimulate similar responses, but together their response is amplified, right? It's like much stronger. So over here you have both the green and the blue that together increase the response, right? So for example, epinephrine and norepinephrine together greatly increase heart rate. Estrogen and progesterone together greatly increase uterine activity. Permissive interactions occurs when one hormone is required for another, right? So the first hormone allows the action of the second. So for example, epinephrine relies on thyroid hormone to produce an effect, right? Some growth hormones rely on cortisol to produce its effect. So you need them kind of, they're in a chain, okay? Um, then we have, I guess you could think of permissive effect like the hypothalamic pituitary axis, right? You need thyroid stimulating hormone to make thyroid hormone, right? Antagonistic hormones create responses that work against each other and oppose each other. We know many of these. Blood glucose, right, is increased by insulin but decreased by glucagon. Blood calcium levels are increased by parathyroid hormone but decreased by calcitonin. So, a patient's this is a little question. A patient's blood glucose levels are measured after administering various cocktails of hormone. What kind of hormone interaction is occurring between these? So, over here you see cortisol, right? You see their blood glucose levels stay constant. You see glucagon, there's a spike and then it goes down. Epinephrine, there's a spike and right. If you bind together epinephrine and glucagon, you get this even higher spike, right? If you bind together cortisol, glucagon, and epinephrine, look at the spike of blood glucose, right? What kind of interaction would this be? You could pause the video and think about it. This is most likely a synergistic effect. These hormones work in synergy to cause an even more amplified response, right? All right, last two topics. Target cell sensitivity. It gets even more complicated. Not only do you have different receptors, not only do you have different hormones, not only do you have different synergies and antagonisms, right? But on top of it, a cell could become more or less sensitive to that hormone, right? To be sensitive, right, means that if a little poke were to touch my hand, ow, I would say that hurts. If I'm not sensitive, then you punch my hand and I say that didn't hurt, right? So if a bit of hormone is put and it could detect it, that's very sensitive, right? But if a lot of hormone is there and it still can't detect it, that's insensitive. And how do we change a cell's sensitivity? Through the number of receptors that they put on their cell surface, right? If they have many receptors there, then they could detect it well. If they don't, they can't detect it well. So over here, upregulation, right? You could change a cell's sensitivity through upregulation, right? If you have persistent low levels of hormone, right? Over here, look at this. In the blood, there's this normal level of hormone, but if you have low levels, right? Persistent low levels, you see less triangles in the blood here. Then over time, right? The hormone causes the target cells to express more hormone receptors, right? Then you put some more hormone receptors on, and the increased sensitivity compensates for the decreased level of hormones. We decrease the amount of hormones, and then the cell became more sensitive, right? Let me give you an example. If you ever have a coffee detox, right? There's a method to that madness, right? Over the detox, you'll be very grumpy and all, but you're gonna deprive yourself of the caffeine, right? And then the cells are like, oh, there's not really much caffeine in the body, right? And so all those receptors are not going to see a lot of this caffeine. And then when you have that first coffee after your detox, then you'll be super sensitive to that caffeine and you'll probably feel the effects a bit more because there was such a lack of it that the cells were saying, where's this caffeine gone, right? So they upregulated a lot of receptors. Um, the opposite, right? Prolonged exposure to high levels of hormone cause the target cell to express less hormone. Here's a lot of hormone, right? And this, right, will kind of overflood the cell and say, look at this, look at this, look at this hormone, right? Over and over. And then the cell eventually is like, I see this all the time. Why the heck do I always need to respond to this hormone? They'll take down some receptors because of this high exposure, right? So the decreased sensitivity compensates for the increased level of hormone, right? This explains why, um, you know, for instance, coffee, if you keep drinking it, right, if you have 5 cups a day, eventually you won't really feel its effects on the body because eventually the cells will be like, okay, I understand there's a lot of this caffeine around. I'm not going to respond to it all the time, right? So that's an example of down regulation. A decreased sensitivity compensates for the increased level of hormone. The best example of this medically is type 2 diabetes, right? Not type 1. Type 1 is a complete inability to make insulin, but type 2 is insulin resistance. So normally, glucose, right, this glucose intake leads to the pancreas releasing insulin. Insulin then binds onto the insulin receptor on these insulin sensitive cells. And these cells could be maybe muscle, adipocytes, right? And then this leads to GLUT transporters. You see these GLUT transporters being recruited onto the cell membrane to allow glucose to enter the cell, right? Now, over here, right, if you have long periods of hyperglycemia, meaning long periods of elevated glucose, right? Throughout your life, you eat a lot of sugar over and over, or fats, whatever that may be, right? But if you have hyperglycemia, constant high amounts of fats, this is a chronic humoral stimulus, right? That will trigger the beta cells of the pancreas to chronically release insulin. Throughout your life, you're going to keep releasing high amounts of insulin because you eat a lot of these sugars, right? This is called hyperinsulinemia, right? You have a lot of insulin being released. As a result of persistently high insulin that's released, insulin sensitive cells like muscle and fat, they will down-regulate the number of insulin receptors, right? It saw a lot of insulin throughout its time, and so eventually it's like, I'm going to stop responding to this, and it will actually only leave a few receptors, so it'll become what we call insulin resistant. Even though there's a lot of glucose, even though there's a lot of insulin, the cells won't want to respond to the glucose anymore, right? Okay, so last thing is the stress response. These are general adaptation syndrome. So this is a series of predictable physiological responses to any type of stress and includes 3 major phases: the alarm phase, the resistance phase, and the exhaustion phase, right? So phase 1 is the alarm, fight or flight. This is our typical fight or flight. The sympathetic response comes first in a stress response. where energy reserves are mobilized and the adrenal medulla releases epinephrine, the dominant hormone, right? All of which cause short-term responses, increasing mental alertness, increases metabolism and sweating, increase heart and respiratory rates and muscle tone, decreased digestive and urinary activity, right? This is a typical fight or flight stress response in the first few seconds, minutes, right? If stress persists for a few hours, right, then you get into the resistance phase. Phase two, you mobilize non carbohydrates, right, including fats and proteins. And this occurs as the adrenal cortex produces the glucocorticoids, right? Remember those glucocorticoids, and the mineralocorticoids, all of which will cause long-term responses. You get to break down adipose stores and skeletal muscle, right? You start gluconeogenesis in the liver to make more glucose for your brain. You will conserve salts and water, right? All of these things occur due to this stress. And the last phase is exhaustion. So if stress persists weeks, months, hormones production and lipid reserves are exhausted, right? So you have no more of those hormones or lipids. Electrolyte levels are unbalanced as sustained aldosterone levels have wanted to conserve sodium at the expense of this potassium. This leads to organ damage, organ system failure, and without correction, your organ failure results in death, right? So this is bad. Review. What should you keep in mind? Hormones are transported by the circulatory system and interact with target cells by binding receptors, either acting directly by crossing that plasma membrane or indirectly by stimulating secondary messengers. Hormones are also categorized chemically as amines, proteins, or steroids. Hormones can act antagonistically, synergistically, or permissively in the body. General adaptation syndrome describes a series of body responses to stress that occur acutely and chronically. So that is it for this lecture.